Hello, everybody. My name is Nathan Greenslet, and I am a second-year master's student in the Department of Marine Sciences, and I work with Dr. Aaron Lipp in Environmental Health Science. So some background on myself. I'm from South Carolina. I grew up in Charleston, and I went to Clemson University for my undergraduate, where I got my degree in biology. It was during my time there that I had my first exposure to research. I worked with a graduate student in a biomechanics lab, and we looked at how animals move on inclined substrates. So I got to work with American alligators as well as stinkpot turtles. It was a lot of fun. But after that, I knew that I wanted to pursue research, but maybe more towards a marine route. I grew up on the coast, so I wanted to continue that. So in December of 2020, I graduated from Clemson and I accepted an internship down in the Florida Keys with a nonprofit called iCare. We did a lot of coral restoration and working with the public and teaching them about restoration practices and coral reef ecology. So it was a lot of diving, a lot of working with people and seeing some really amazing life underwater. This image here on the left is a picture from one of our monitoring dives when we would go back and check on the corals that we put back out onto the reef. And here was a loggerhead sea turtle that was using a sea fan to scratch itself. And then there's a barracuda in the back. So that was just sort of one of the days that definitely stands out to me while I was down there. And it was a lot of fun. Then as my internship wrapped up, I started looking around for graduate programs and I found UGA, specifically Dr. Aaron Lipp, who is an environmental microbiologist interested in water quality and how it impacts both human and marine health. So this past year, I've been learning a lot about microbiological techniques and molecular techniques such as DNA and RNA extraction, culturing, and PCR. So it's been a lot of fun, and I've been learning a lot. Sort of stepping out of that science zone for a second, things that I like to do here in Athens when I'm not doing science is there's a place called Rook and Pond that's basically sort of like a coffee shop or bar. So you can go and have some coffee or drinks with some friends. And then as you can see from this image, they have a wall of board games. So you can just spend the whole day there playing a lot of really fun and interesting games. I also like to run, and the Athens Running Company has a running club that meets every Thursday. So we'll go and hang out and run around Athens. You become really familiar with certain routes. And then they give you some food and drinks afterward, which is always really great. And then also the State Botanical Garden is not too far away from here, about 10 or 15 minutes, and they have some really awesome trails that are fun to go and explore. So diving into what I specifically do, I'm interested in Saharan dust events and how they impact microbial communities in coastal waters. So each year, about 10 to the 8th metric tons of dust from the Sahara Desert is supplied up into the atmosphere. And that dust is carried across the Atlantic via the easterly trade winds. And as it moves, some of it is deposited in the surface waters of the Atlantic, as well as the Caribbean and the Gulf of Mexico. These so-called dust events happen about 3 to 5 times a year and last about 3 to 7 days each. And these dust constituents include a wide variety of bacteria, virus-like particles, fungi, minerals, and nutrients like nitrate, phosphate, and iron. And this image here in the bottom left is a good example of a dust plume being emitted into the atmosphere, carried across the Atlantic. And then by the time it reaches sort of downstream, you have particle sorting. So all these larger pieces sort of fall out and you are left with this fine, potentially more biologically available particulate traveling across, and that's really nutrient rich. And as you may know, when you add nutrients to an otherwise oligotrophic setting, like the Florida Keys, you can have blooms of bacteria or phytoplankton. One such case is harmful algal blooms, which is caused by the Dinoflagellate Carenia brevis. That's been shown to occur in the Gulf of Mexico in response to these dust events. But I'm interested on the bacteria side of things, specifically Vibrio, which is a genus of bacteria. They're gram-negative, rod-shaped, and opportunistic heterotrophs. There's about 100 species or so, and they're pretty much found in any marine environment. If you take a water sample and culture it, you will most definitely find Vibrio there. But they're ubiquitous, but in really small amounts. They can also be found in freshwater environments, but that's more of an exception than a rule. They're free living in the water column. They can also live in association with animals like fireworms, and even have some symbiotic relationships with squid, which is really cool because they can actually fluoresce and create this phosphorescence underwater. Temperature and salinity are really important drivers that impact their abundance and virulence. So they typically like around 20 to 35 parts per thousand, as well as between 15 and 35 degrees Celsius. And these bacteria have been characterized among some of the first to respond to these dust events in waters. And this can pose an issue because Vibrio can cause a wide variety of diseases in humans. Humans can get sick through a waterborne route. So if they have a wound and they're in the water, Vibrio can access the wound and get people sick that way. You can also get sick from contaminated drinking water. In terms of foodborne, you might have heard that you're not supposed to eat things like oysters in the summer months, especially raw or uncooked seafood. And this is because in the summer months, when people harbor oysters, they'll take it and the oysters will close up when they become exposed to the air. And that creates a really nice environment for a lot of bacteria to build up. It's warm and moist. And so you can have this buildup of bacteria. Vibrio can cause a wide variety of diseases, ranging from cholera to gastroenteritis, wound infections, septicemia, and necrotizing fasciitis, or flesh-eating disease in some extreme cases. A subset of species can also pose an issue for marine health. They've been shown to cause bleaching and eventual tissue loss in coral species. So here in this bottom image here, we have a healthy coral. Vibrio were inoculated into the water column, and you can see it's bleached, so it still has its tissue. If you look really closely, you'll see a thin layer of tissue and small little polyp tentacles. But then at elevated temperatures, you'll see that we actually eventually had tissue sloughing to where the actual skeleton isn't exposed. You can also have shellfish mortality, as well as fish mortality. And this is a sea bream here, and you can see that it's had hemorrhaging in the gills. That can pose a lot of issues for foundational species, which are really important for ecosystem balance, as well as a really important aquaculture species like shrimp or fish. So that can ultimately impact us humans as well. So this summer, I was interested in looking in an area, specifically the Gulf of Mexico, towards the end of this dust route. So I traveled to Corpus Christi, Texas, and we did sampling across 3 sites before, during, and after a dust event. And since then, I've been here doing a lot of lab work, a lot of filtration, DNA extraction. Then we have this eluted DNA, and we can use that for quantification using quantitative PCR or qPCR, as well as using PCR, conventional PCR, to look at certain communities, the bacterial community or eukaryotic community, as well as the Vibrio species specific community, and seeing how these things change over this time series. But it's been a really awesome experience, and that's sort of just a quick introduction to what I've been up to and what I like to do here in Athens. But if you ever have any more questions or this sounded interesting to you and you want to talk more, I'm more than happy to set up a meeting or a chat at some point. So here is my email. And yeah, if you have any questions, let me know. But I hope you enjoyed my presentation.